innovated in the last few years in the revision of essential data on biosimilars. So Philippe is much up to date on this theme. And we will have his presentation first. And then, as we have already one presentation and another one, and we still have two from my side, what I was planning, if you agree, is that after Philippe, you can have, like Carolina told you, a bio break. A little bit, a moment to go to the restroom, to have coffee if you want, to drink some water, or whatever you want, to relax a little bit. And then I will concentrate, I will condensate the two presentations in just one, okay? And also adapt to the time that we have left, okay? Instead of having exactly what we have there, in terms of the time the duration of my talk, I will compensate that exactly as I did yesterday, okay? Adapting the slides and making the time schedule equal to what we have in terms of the hour that we finished, okay? So, Philippe, please.
So, good morning, everyone. I would like to start by thanking Professor Fonseca for the kind invitation to be here and present to you some data on rituximab biosimilars. My name is Filipe Araújo. I work at a very nicely located hospital, the Hospital de Santana, which is located in the coastline close to Cascais. And there's a reason why this hospital is so well located. It was created in the beginning of the 20th century because it was used to treat tuberculosis. And so it used the properties of the air and the properties of the sun to treat tuberculosis patients. And so it was later converted to an orthopedic hospital with a nice view, as you can see. This is the view from my office. I'm just trying to make you really jealous. And this is me during my lunch break. So this is an orthopedic hospital. So I'm used to dealing on a daily basis with orthopedic surgeons who usually are very objective and don't have any time to lose. And so I'll try to keep my presentation as objective as possible. These are my disclosures. So this is the schedule for the next 20 minutes. We'll go through rituximab biosimilars approved on the market, and then we'll go through ongoing clinical trials on rituximab biosimilars. Just a couple of ideas on switch and immunogenicity, and also on pharmacoeconomics, and then we will finish. So as for the introduction, as you all know, rituximab is a murine human chimeric monoclonal antibody of the IgG1 type. It's a B-cell depleter. It acts through binding the CD20 surface antigen, and it's thought to cause a depletion of the B cells, at least with 4 different mechanisms: complement-dependent cytotoxicity, antibody-dependent cytotoxicity, Fc gamma receptor-mediated phagocytosis, and induced cell death. Rituximab was first approved for the treatment of follicular lymphoma in 1996. It was later approved for the treatment of other hemato-oncologic diseases. And then in 2006, it was approved for the treatment of rheumatoid arthritis in patients who were at least intolerant or inadequate responders to at least one TNF blocker. It was later, in 2011, approved for the treatment of small vessel vasculitis, namely granulomatosis with polyangiitis and microscopic polyangiitis. However, we do use it in other indications on a daily basis. We have several off-label indications, usually in severe or refractory organ involvement in these rheumatic conditions. As you have been seeing in the past 2 days, that we do have an explosion of biosimilars ongoing. We have several approved biosimilars in highly regulated markets that have been already approved, and we have many other biosimilars in the pipeline in different stages of development. That's true for infliximab, etanercept, adalimumab, and also for rituximab. We have currently 2 approved rituximab biosimilars in the European market, namely CTP10, which is Celltrion's biosimilar, brand names Truxima, Blitzima, Ritemvia, and Rituzena. And we have another biosimilar approved, GP2013, which is Sandoz biosimilar, brand name Riximio and Rixaton. We don't have any approved rituximab biosimilar in the United States, although these two pharmaceutical companies have submitted a BLA, a biologics license application, that we have recently heard were rejected, both of them. The FDA rejected Sandoz proposal and also rejected Celltrion's proposal for these two biosimilar candidates. We still do not know the reason for the Sandoz biosimilar refusal, but we do know that refusal of Celltrion's biosimilar was related to an inspection performed by the FDA in last year, 2017, where they supposedly found some poor aseptic conditions in the plants in South Korea. So let's go currently approved biosimilars on the market. As you all know, all biosimilars that are approved have to go an extensive, intricate totality of evidence package. They have to deliver it to the regulatory agencies. I will not go through the preclinical assessment because that would take me a whole day, and Professor Gonçalves has explained it briefly, all the analytical and functional processes. But you do have here the reference for an extensive analytical assessment of CTP10. Regarding the phase 1 trial, the phase 1 trial was performed in 154 RA patients who were inadequate responders or intolerant to at least one TNF blocker. It was a 24-week randomized controlled double-blind trial. This was a trial that was intended to prove a pharmacokinetic similarity between CTP10 and originator rituximab, and indeed both primary pharmacokinetic endpoints were met. Both the area under the curve and the concentration max, the confidence intervals of the ratio of geometric means, they were within the bioequivalence, pre-specified bioequivalence range. This study also assessed other outcomes, other endpoints, namely efficacy and safety endpoints. As for efficacy, we can see that the ACR responses, ACR 20, 50, and 70 responses were very similar between CTP10 and the bio-originator. And that was also true for the EULAR, good, and moderate responders. In terms of safety, I don't have it here, but safety was also similar between CTP10 and the bio-originator in terms of treatment emergent adverse events, treatment related adverse events, serious adverse events, infections, infusion reactions, and malignancies and deaths. We have results at 72 weeks in terms of efficacy, and as you can see, the phase 1 results at 24 weeks, they persist throughout the 72 weeks in terms of clinical efficacy and also clinical safety. The phase 3 trial of CTP10 was a trial that was performed in 372 RA patients who were also inadequate responders or intolerant to at least one TNF blocker. And we can see here that the responses, the primary efficacy endpoint, which was the difference in terms of DAS28 response, the confidence intervals were well within the pre-specified range, which was in this case minus plus 0.6. The same thing for the ACR responses and the EULAR responses. And also the same thing for safety. It was similar between both treatment arms. There's also a phase 3 trial of CTP10 in patients with advanced stage follicular lymphoma, meaning Ann Arbor stage 3 or 4. And as you can see, the primary efficacy endpoint, it's not very clear because the letters are really small, but you can believe me, the efficacy endpoints were met and they were within the pre-specified bioequivalence range. The same thing for the pharmacokinetic endpoints. As for GP2013, this is Sandoz biosimilar. You have here two references you can check for the preclinical assessment. Regarding the clinical assessment, the phase 1 trial was performed in 302 RA patients who were also inadequate responders or intolerant to TNF blockers, and they were assigned to either the biosimilar or United States sourced rituximab or European Union sourced rituximab. Once again, the primary endpoints of pharmacokinetic nature were met, all of them, the area under the curve and also the concentration max. And you can see here some efficacy endpoints which were also met. And the same thing for safety once again. The phase 3 trial was performed not in rheumatic patients, but in hematologic patients. It was performed in patients with follicular lymphoma and Arbor 3 and 4 classification who were treated with either the biosimilar or originator rituximab in combination with the CVP protocol, which is usual in this type of follicular lymphomas, meaning cyclophosphamide, vincristine, and also prednisone. And once again, we are not used to these efficacy endpoints, but the endpoint was an overall response, an overall tumor response, which was the primary endpoint was met, because the difference was minus 0.4%, and the confidence intervals were within the the pre-specified equivalence range of minus plus 12%. What do we have in terms of ongoing clinical trials of rituximab biosimilars? I don't want to be too much extensive. I just want to say that Pfizer has a biosimilar that's currently being developed. It's called PF0528056, which is pretty much unpronounceable, so I'll just call it Pfizer's biosimilar. We have data published as a full paper in terms of preclinical assessment and also in terms of phase 1 trial. We don't have yet phase 3 trial results, but they have been announced to be positive, and they were performed in patients with follicular lymphoma. And there are also naturally extension trials of the phase 1 and phase 2 currently ongoing. We also have an Amgen's biosimilar, ABP798, with only known preclinical data available. They were presented in the European Congress of Hematology, and we don't have yet results for the phase 1 or phase 3 trials, although they have been completed. Boehringer Ingelheim had a biosimilar of rituximab whose development was suddenly stopped in 2015 after the completion of the phase 3 trial. It was said to be, the development was said to be halted, not due to safety or efficacy issues, but due to policy of the company, which is a bit strange considered it was halted immediately after the completion of the phase 3 trial. As you do know, we have some intended copies of rituximab that have been used for the past years in non-regulated or at least less regulated markets. We have intended copies of rituximab, namely Novex and Kikuzubam, which are used in some countries of South America. We have Aselbaya, which is an intended copy of rituximab currently in use in Russia. And we have the remaining that are currently being used in India. Just a couple of ideas on switch and immunogenicity. Data on switch are very limited. We only have 3 trials that have assessed immunogenicity. 2 of them are extensions of the phase 1 trial. The first, the phase 1 open label extension of CTP10, assessing a single switch in a very limited number of patients. These are the patients, it's a low number of patients because these are the patients that needed a second or a third course of treatment with rituximab. So we have a low number of patients, but we have positive results, comparable efficacy and safety after switch. The phase 1 extension trial of Pfizer's biosimilar kept the randomization and kept the double blindness. It was a single switch trial with also a low number of patients, but with comparable pharmacokinetic pharmacodynamic and safety endpoints met. A specifically designed to assess a switch trial was the GP 2013 trial, in which patients who had been previously treated with the bio-originator were randomized to receive the bio-originator or to receive the biosimilar. A low number of patients, but with comparable safety and immunogenicity after switch. I don't want to go through the immunogenicity issue because Professor Gonçalves has made that in a beautiful way. I just want to say that all trials, basically all trials, assessed immunogenicity, and we have similar percentage of patients developing anti-drug antibodies, both for rheumatoid arthritis and follicular lymphoma. As you can see, patients with follicular lymphoma have a less incidence of anti-drug antibodies, and that's true for CTP10, GP 2013, and Pfizer's biosimilar. In terms of pharmacoeconomics, as you know, rituximab is a top-selling drug in the world. It's a top-selling drug. It's a bestseller for Roche Pharmaceutical. Last year, in 2017, the sales for rituximab were approximately $7.9 billion worldwide. So there's great expectation as the cost savings that this biosimilar may bring. And there's a very beautiful budget impact analysis published last year on CTP10. It was a budget impact analysis that had a budget impact model built on 3 assumptions. In 28 European countries, assuming a biosimilar consumption, a rituximab consumption, 18% RA patients, 72% oncologic conditions, and 10% probably off-label indications, and assuming also that the biosimilar would cost 70% of the bio-originator's price, assuming over 1 year that there would be a 30% uptake of the biosimilar, that would mean savings in the order of 90 million euros. And that would also mean that 7,500 additional patients would be treated. Considering a more optimistic scenario of 50% uptake over 1 year, that would mean 150 million euros in savings and also almost 13,000 additional patients treated. A 3-year model assuming a 20, 30 and 40% of uptake in the first, second and third year would mean savings of 570 million euros and almost 50,000 additional patients treated with rituximab. So, to conclude, I would like to say that rituximab is an important asset for the treatment of rheumatic conditions and also hemato-oncologic conditions. There are 2 biosimilars approved in the European Union and 2 are currently under development. Data on switch are very limited, but safety and efficacy don't seem to be affected after a single switch. The uptake of biosimilars of rituximab will offer significant cost savings and improved access to both rheumatic and hemato-oncologic patients. And that's it. Thank you for your attention. Passa-me aí a água. Desculpa, Pedro. Água, desculpa. Não, nada. Passa-me só a água. Obrigado.
Sobre la situación de rituximab. Yo creo que vamos construyendo un caso de fármaco por fármaco, especialidad por especialidad. Yo creo que esta presentación, y con los hematólogos que deciden, debería ser una presentación que levantara un cierto nivel de preguntas. Así que yo creo que las podemos hacer en español, ¿verdad?
Sí. Perfecto.
No hay preguntas.
Foi claro demais.
Creo que sí, pero Simon había hablado del hospital decían con un 90% de entrada, 95%, y ellos solos tenían 4 millones de ventas. Luego, el escenario económico de ahorro me parecía pequeño. ¿Por qué no ha entrado tanto el de Sandoz?
Provavelmente porque foi aprovado depois. Probably it was because it was approved later, considering to CTP10. Mas é uma pergunta difícil de responder. Há inúmeros fatores que condicionam a entrada de um biosimilar. E os fatores são diferentes de país para país, de mercado para mercado, de hospital para hospital. Em Portugal, cada hospital tem uma forma de negociar diferente e cria muita heterogeneidade. Não há prova de que o biosimilar da Sandoz seja pior, mas provavelmente existem outros fatores que condicionam a melhor penetração de um comparando com o outro.
E neste hospital, qual é a penetração do rituximab biosimilar?
No meu hospital, zero, por enquanto. O biosimilar do rituximab chegou há muito pouco tempo a Portugal. Já tem cá algum, Pedro? Aqui também não há. E este é um hospital...
É uma condição que é muito diferente para a utilização do rituximab em patologia reumatoide ou autoimune e em hematologia. En hematologia temos muito menos evidencia de efectividad, mucho menos tiempo de observación y también los escenarios completamente diferentes, porque otros usar un anticuerpo monoclonal en una situación autoinmune, otros usar un anticuerpo monoclonal en situaciones de déficit de inmunidad. Y segundo, el target es diferente, porque en la hematología, en los linfomas, el target es la célula, nell'arteria reumatoide il target è nell'anticuerpo, entonces cosas muy diferentes. En los Estados Unidos no tiene ningún biosimilar aprobado, pero ninguno, no solamente uno. ¿Por qué?
De rituximab?
Sim.
Não. Os Estados Unidos têm tido um maior atraso na aprovação de biosimilares. Aconteceu o mesmo com o biosimilar do infliximab, CTP13. Eles têm standards que são que são diferentes, não substanciais. Segunda vez.
O segunda ou terceira vez, que já lo bloquearon.
Detalles no lo sé, yo también me quedé un poco sorprendido porque desconocía el de Sandoz.
E as razões são desconhecidas. A Sandoz não disse porque é que foi rejeitado.
No, tiene una publicación ahora que recién, muy recién, tiene una publicación donde tiene una explicación, pero no muy comprensible, no muy
Pero
sólida.
también entienden, no estoy hablando de proceso de aprobación, pero primero los Estados Unidos tienen una medicina mucho más litigante que la nuestra. Entonces, normalmente, en algunos aspectos, la presión farmacéutica es menor porque allí el grueso de la tostada se la lleva el sueldo del médico. Es decir, en Estados Unidos, recuerden que tienen hasta la figura del physician assistant, que es un señor que simplemente intenta retrasar al máximo que un médico, su tiempo que es muy caro, sea utilizado. Por tanto, ellos, el énfasis está más balanceado que los sistemas nuestros europeos, donde el salario del médico es fijo y es el coste del fármaco con población además que está envejeciendo y es generalizado, que se lleva mucho como es eso. Pero luego recuerde que hay temas de patentes, por ejemplo, la adalimumab aquí va a caer en octubre, pero en Estados Unidos va a durar 2 o 3 años más. Entonces, y hasta ahora tienen una política que había estados, que no sé si ha cambiado, donde estaba hasta prohibido pelear por un descuento del precio, pero por ley, porque eran lobbies muy fuertes, ¿de acuerdo? Yo hasta ahora pensábamos, yo eso no lo atribuyo para nada al rechazo del fármaco de esto, porque esto es la FDA y esto no tiene nada que ver, ¿no? Pero sí que yo personalmente cuando le he oído decir eso voy a explorar ese tema.
O ano de queda da patente também é diferente na Europa e nos Estados Unidos, como sabem.
En el mundo de los biosimilares que vemos bien, ¿verdad? La política de farmacovigilancia e instauración de métodos de inmunogenicidad, ¿qué nos llevarían a cambiar la práctica clínica? La práctica clínica, no en la investigación.
¿Alguna
A ver, yo creo que
nada?
aquí nos está faltando todo un tema, que es un registro de la farmacovigilante que la logrará el doctor Fonseca. No es tanto en términos de determinación de los médicos que no los tuyos, sino en términos de trazabilidad de un paciente, ¿de acuerdo? Y del lote de fármaco que está llevando un paciente, que vamos a necesitar una gran conexión entre el farmacéutico y el médico, y un registro de los pacientes. Hasta ahora la EMA exigía 2 años, sugería 2 años de control, cual incluso en los primeros momentos de entrada de los biosimilares fue utilizado para hacer cambios de precios en algunos hospitales. Entraban en un hospital muy barato, conseguían el hospital y luego les decían, ahora no me puedes cambiar porque tienes que tenerme vigilado 2 años. ¿De acuerdo? Pero eso yo creo que ya no está cambiando. Pero que ustedes han visto al profesor farmacéutico inglés, él ya estaba anticipándose un escenario del múltiple switching. Y lo que le habían dicho es, tú entras ahora y durante 2 años no puedes cambiar. O sea, si sale un fármaco más barato, no te voy a exigir que cambies, no te voy a penalizar. Pero eso es un sistema que nosotros no disponemos, porque en España y en ustedes los hospitales se mueven prácticamente de forma muy independiente. De hecho, en mi país está casi muy cuestionado la compra para grandes sistemas porque las compañías se sienten excluidas. Como van por puro precio y los análisis de calidad son muy bajos, nos hemos encontrado, por ejemplo, en Andalucía, que todos dicen que ha sido rechazada porque ninguno de los fármacos que se compró, yo conocía el laboratorio que lo vendía. Se le sirve como ejemplo, es decir, si laboratorios que yo nunca había oído hablar de ellos fueron los que se llevaron y salió una cosa muy rara. Aquí hay un intercambio entre ciencia y mercado, ¿de acuerdo? Y yo creo que lo que vamos a tener que hacer es asumir que cuando hayan 3 nuevos, mantener los nuestros durante algún tiempo hasta que generemos un cierto nivel de experiencia. Si empezamos a cambiar, terminaremos teniendo una política de eso. Yo cuando estaban hablando de niveles plasmáticos, yo sé que muchos de ustedes, en algunos hospitales de ustedes, no tienen la posibilidad de regular el tratamiento con niveles plasmáticos, que aquí es bastante habitual. ¿De acuerdo? O sea, nosotros controlamos nuestros pacientes cuando fallan con una determinación de niveles plasmáticos de anticuerpos. Entonces podemos, pero eso ha costado tiempo, pero eso es un paso para ustedes, ya no para el biosimilar, sino en la optimización de resultados suyos.
Muy difícil esto, es imposible hacerlo en todo el hospital. A menos que no se haga un estudio randomizado, controlado.
Práctica.
Sí, en práctica es imposible hacer. Pero si no tiene la farmacosurveillance muy bien hecha, no se puede hacer.
A ver, imposible solo es ponerse a empujar un poquito, estas técnicas tampoco son tan sofisticadas, ¿de acuerdo? O sea, no es, costó, yo vi la entrada de este, costó, pero una vez entra, el ahorro que tú consigues porque puedes optimizar y mover a tus pacientes según los del tratamiento también cuenta, ¿eh? Eso, a veces haces tratamientos que ya están fallidos porque no estás midiendo los niveles plasmáticos y aquí los puedes...
Aparte del fatto que no se sabe todavía, por ejemplo, en el rituximab y en patología no se sabe cuál es el nivel plasmático optimal que uno tiene que obtener, porque uno tiene un nivel plasmático de 25, uno de 10, el resultado es lo mismo, entonces...
No estoy discutiendo, también me voy a disculpar, yo tengo un problema, yo soy más del campo de la reuma y del campo del gastro y ahí, claro, ahí mi creencia es por eso, ya sé que con ustedes es diferente. ¿Alguna pregunta más? Pues vamos a hacer 5 minutos de parada técnica y acá.